Hi students, today we are going to discuss one more important topic from lipid metabolism. Essay question, short notes, MCQ, everything, clinical scenario, right? Very important essay question, right? I already told you ketone body metabolism is the first important essay question from lipid metabolism. The second one is beta oxidation of fatty acids, right? We have oxidation of fatty acids four types: alpha, beta, gamma and omega. Beta is your essay question, alpha, gamma, omega is coming under your short notes, right? So today the essay question, beta oxidation. You all know why it meant for beta oxidation, means the cleavage site is beta carbons, right? So after removal of two carbon units, so successive removal of two carbon units in the long chain fatty acids. What is the concept for this beta oxidation of fatty acid is, first you have to take one 16 carbon long chain fatty acid. After beta oxidation, they will go for successive removal of two carbon atom. That two carbon atom is called as acetyl-CoA. So each and every cycle, because this oxidation takes 7 cycles per oxidation. So each cycle it will release 1 2 carbon molecule, that is acetyl-CoA. Finally one more acetyl-CoA. So totally 8 acetyl-CoA released from this beta oxidation. So the concept is one long chain fatty acid which will successively removes 2 carbon atoms and finally produces last 2 carbon atoms. All the acetyl-CoA enters into TCA cycle for the production of ATP, right? Acyl-CoA produces means which will again come for the next reaction. I will explain in the diagram. For this beta oxidation, it actually so the oxidation occurs in all cells. Actually there is one MCQ for you. These are all the organs which is not having oxidation of fatty acids because of the absence of the oxidation enzymes, that is brain, RBC and adrenal medulla, which is not able to oxidize fatty acids for their energy. Okay. Second one is, the compartment for this beta oxidation is both cytosol as well as mitochondria, right? We will briefly discuss in the coming discussion. That is, first activation takes place in the cytosol, then the molecule enters into the mitochondria where the oxidation reactions will happen. Okay. So the first one is oxidation of fatty acids. So after fatty acid synthesis, that is lipogenesis, some free fatty acids depending upon the situation enters into beta oxidation. That is, lipogenesis is over means, or depending upon the situation, the fatty acids will go for oxidation. Then only they will produce some amount of energy, right? So how it will get activated? First you will take fatty acid, that is for example palmitic acid. It is a 16 carbon long chain fatty acid. Okay. So from the 16 carbon long chain only we are going to cut 2 carbon atom of acetyl-CoA, right? So this fatty acid converted into acyl-CoA, that is activated fatty acid. This is the first step. Fatty acid, that is palmitic acid, which is converted into acyl-CoA, right? For this conversion we need, this is the enzyme, this is the cofactor, that is the enzyme thiokinase. Another important MCQ, another name is acyl-CoA synthetase. Synthetase means we are utilizing 2 molecules of ATP. So the enzyme name is acyl-CoA synthetase or thiokinase. Energy, we are giving 2 ATP molecules for the activation of this process. Cofactor is magnesium and we are adding one more coenzyme. For beta oxidation, 2-3 places coenzyme involvement is there. Don't forget to mention about the vitamin CoA, right? So this coenzyme SH, that is CoA, ATP thiokinase for the conversion of acyl CoA from fatty acid. This is the number one reaction, activation of fatty acid. Second one is, this happens in cytosol. Actually, we want to have the reactions in mitochondrial matrix. So there is a membrane. This membrane is not permitted the acyl CoA to penetrate into the matrix. So we have a transporter that is called carnitine shuttle. Through this carnitine only, the acyl CoA enters into the membrane and goes to the matrix. How? This acyl CoA, already we have acyl CoA, combines with carnitine, converted into acyl carnitine and CoA. This is the first reaction, right? This is the first reaction. Here the enzyme is CAT1, that is carnitine acyl transferase 1. This acyl carnitine is now permeable to the membrane and goes to the mitochondrial matrix, whereas acyl CoA is not permeable to the membrane. That is very, very important point. So this acyl carnitine enters into the membrane through the translocase, which is the transporter present in the membrane. Okay. So in the matrix side, acyl carnitine is ready for the third reaction. This is actually second reaction. Third reaction, this acyl carnitine combined with another CoA coenzyme converted into carnitine and acyl CoA. That is, it releases carnitine for the another reaction, and we now have the acyl CoA for another oxidation process, right? So this acyl carnitine combined with CoA produces acyl CoA and carnitine. This carnitine is again for the next reaction coming to the cytosol. Whereas in the matrix, what happens to this acyl CoA? This is actually third reaction, this is the fourth reaction. Totally 4 reactions for the transportation of CoA, right? Acyl CoA. So in the matrix, this acyl CoA go for the oxidation, beta oxidation reactions, actually 4 steps. After that we will have acyl CoA and acetyl CoA. This acetyl CoA enters into the TCA cycle and produces ATP, right? So first point is activation of fatty acid. Second point is transportation through the carnitine shuttle. Okay. Now we are moving to the reactions of beta oxidation. This is very, very important point. We have 4 reactions, 4 steps with 4 enzymes. All the 4 enzymes are very, very important because they will have some energetic value. They will produce some amount of ATP. So you have to write down the steps. You have to mention the name of the enzymes. No other way, right? So here the names, because of my experience, I am telling one by one very frequently, easily. For you it is very tough, but you have to read all the 4 names. Fatty acyl CoA now enters into mitochondrial matrix, which is available for the oxidation reactions. This fatty acyl CoA converted into trans-enol CoA, then hydroxyacyl CoA, then keto-acyl CoA, finally acyl CoA and acetyl CoA. 5 names you have to memorize. Fatty acyl CoA, trans-enol CoA, hydroxyacyl CoA, keto-acyl CoA, and finally releases acyl CoA and acetyl CoA. What are the important points in this 4 is, first dehydrogenase, second hydratase, third dehydrogenase, fourth thiolase. Thiolase is the cleavage step where it will give 2 products. Whereas this one is dehydrogenase, means you know the oxidation and reduction reaction means the enzymes are, vitamins are riboflavin and niacin. Here we are using one FAD, that is riboflavin coenzyme active form, which will be converted into FADH2. In biochemistry, anywhere dehydrogenase involved FAD means they will produce FADH2. automatically enters into respiratory chain or electron transport chain for the production of ATP. Next one is hydratase, here nothing specific. Third one is again one more dehydrogenase. Here we are using NAD, niacin coenzyme, active form of niacin NAD, which will be converted into NADH and H+, automatically enters into a respiratory chain or electron transport chain for the production of ATP. The last one is thiolase, which is cleaving the keto-acyl-CoA into acyl-CoA and acetyl-CoA with the help of one more coenzyme. That is why I already told you coenzyme is very, very important for the beta oxidation step. Okay, what is the fate of these two products? We know acyl-CoA again enters for the oxidation reactions, whereas acetyl-CoA enters into TCA cycle. Usually, normally what happens is anywhere in our body acyl-CoA producing means they will be entered into TCA cycle for the production of ATP. So this is the beta oxidation steps for your essay question. Very, very important steps, right? You know FADH2 produces some 1.5 ATPs, NADH produces 2.5 ATPs, acetyl-CoA through TCA cycle produces 10 ATPs. We will be discussing in energetics topic because energetics of beta oxidation is very, very important short notes, right? The last one is regulation. How this beta oxidation is regulated? We have some 4 points. One is the availability of the substrate. That is, after lipolysis, the free fatty acids available means they will enter into mitochondrial membrane for the oxidation. Otherwise, it won't happen. So, availability of the fatty acid. Second one is activity of the CAT1, that is carnitine acyl transferase 1. In the carnitine shuttle, we have two enzymes, CAT1 and CAT2. But CAT1 is the regulatory enzyme for beta oxidation. This only allows the fatty acid into the mitochondrial matrix, right? Second one is you have to mention about the malonyl-CoA. This is actually fatty acid synthesis enzyme. If this will be predominant means this CAT1 will be inhibited. That is, synthesis only occurs, whereas oxidation absent. If oxidation occurs means synthesis will be absent. Like that, it will go parallel. Last one is routine hormonal involvement. Actually, here the enzyme is CAT1, that is the pathway enzyme. Hormonal regulation means insulin always decreases beta oxidation, whereas glucagon stimulates the beta oxidation because glucagon only synthesis stimulates the lipolysis. That is the only hormone which will be giving fatty acids to the beta oxidation. That is why glucagon stimulates beta oxidation, right? Last point for this essay question, you have just mentioned about the disorders involved in beta oxidation. Usually carnitine deficiency, that will produce some muscle cramps, muscle pain, and sometimes absence of fatty acid leads to overutilization of the glucose. The patient will go for hypoglycemia also. These two are the important MCQ related to the disorders in beta oxidation. Okay, very, very important essay question. Activation of fatty acid, transportation of acyl-CoA, beta oxidation reactions, right?